Thank you, Dr. Han, and thank you for your for the introduction. I come from Yat-sen University in Guangzhou, China, which was called Canton in the ancient times. It's a great honor for me to be invited to this conference. So today I represent my supervisor, Professor Wei Su, who is a famous expert in the field of traditional Chinese medicine, to give this oral presentation. So let's talk about naringin first. Naringin is a dihydroxyflavonoid compound. It distributes widely in rutaceous plants such as pomelos, lemons, oranges, and so forth. The naringin used in the study was extracted from traditional Chinese medicine, Huaju Hong, which is a geoauthentic root herb in South China and used as an excellent antitussive and expectorant for more than 1,600 years. Huaju Hong was also listed as a royal distribute in Qing dynasty in ancient China. It is the dry epicarp of the young rutaceous plants called Citrus gradis tomentosa. The modern pharmacological researches showed that naringin has the effects of, this is the aglycon form of naringin. It is called naringenin. It has antioxidant, anti-inflammation, antitussive, and so forth, these pharmacological effects. As we know, it is difficult to screen out the most effective leading compound from traditional Chinese medicines with traditional chemical screening strategy like bioactivity leading isolation or chromatographic fingerprint method. The bioactivity leading isolation is labor-intensive and time-consuming. And the chromatography method is with bare connection with bioactivity information. So we first developed a method to solve this problem. We call it fingerprint efficacy relevance analysis. We first collected batches of Huaju Hong samples and extracted them with water and analyzed the chemical components of the extraction and got the HPLC fingerprints for each Huaju Hong sample here. This provides the chemical information of the mixture. At the same time, the extraction were also tested by animal models of cough to quantify the antitussive effects of different samples. So we found that the coefficients of substance naringin was always the largest in different analysis methods. And its aglycon naringenin has large positive coefficients too. These results suggested that naringin is the most essential substance contributing the antitussive effect of Huaju Hong. For naringenin, even though its coefficients were large, but its content in the Huaju Hong is 1,000 times less than naringin. So it is too less to be a future drug. After screening the active ingredients, we have extracted naringin for Huaju Hong. The purity reached 98.8% and determined the pharmacokinetic parameters in rats and dogs. Because of naringin's rapid transforming into naringenin and conjugating with glucuronic acid, we freed all the naringenin from its glucuronic form in plasma with beta glucuronidase. We measured the total naringin, including free naringenin, and the naringenin free from the conjugation. To describe a concentration time curve in order to reflect the efficacy of certain components, naringin and naringenin. So we could see the pharmacokinetic parameters in rats shown in this table, and we could see it is the AUC was linearly associated with dose. This is the linear equation. and this is for dogs. This is the linear equation. Similarly, the bioavailability was 46.11% in rats and 36.82% in dogs. So naringin can be absorbed rapidly. The blood concentration reaches the Cmax rapidly and eliminates rapidly. It has a short half-life period and moderate bioavailability. According to the metabolism study, naringin transformed into its aglycon naringenin and conjugated with glucuronic acid. So the sample should be hydrolyzed and measured, the total naringin. Okay. In order to reflect the in vivo cause of the extract effective substances. We also studied the tissue distribution of naringin in rats. Samples were dealt with beta-glucuronidase and extracted using organic solvents. Endogenous matrix in different blank tissues samples from 6 individual rats were evaluated. Following an oral administration of 42 milligram per kilogram naringin to rats, the tissue concentrations of naringin and the total naringenin determined at 7 time points are tested. The naringin concentration were high in stomach, small intestine, liver, and trachea. The high levels of total naringin were found in liver, stomach, small intestine, kidney, lung, and trachea. After an oral administration of naringin to rats, both naringin and total naringenin were widely distributed to all tissues tested except brain. The undetectable levels of naringin and total naringenin in brain suggested that the polarities disabled them to cross the blood-brain barrier. Moreover, naringin and total naringenin were few or undetectable at 12 hours. At 12 hours in all tissues examined, which suggested that there were no long-term accumulations in the tissues. These characteristics were similar to our previous reports in rat plasma. So the present study demonstrated that naringin is rapidly and widely distributed to most tissues but cannot cross the blood-brain barrier in rats. There are no accumulations in tissues of rats. And this smaller figure is the detail of these tissues, liver, kidney, trachea, and so on. Well, we could see that in brain. In order to study the form of naringin in systemic circulation, we also study the protein binding ratio of naringin and naringenin in different species. As we know, equilibrium dialysis, microdialysis, high performance affinity chromatography, and so on, all these methods could be used to study the plasma protein binding ratio. Compared with other methods, equilibrium dialysis has simple devices and steady protein binding. In our study, the rat, dog, human plasma were used. Plasma containing naringin and naringenin, naringin from 100 to 5,000 nanogram per milliliter, or naringenin from 100 to 10,000 nanogram per milliliter was used. The time to reach equilibrium was tested by collecting samples from both buffer and plasma at 2, 4, 6, 8, 12, and 24 hours. So plasma protein binding of naringin and naringenin was assessed after incubation at 37 degrees, within equilibrium time achieved. Plasma and phosphate buffer samples were collected and assayed by LC mass system. So we could see after 6 hours, the plasma concentration of naringin, naringenin, and buffer concentration of naringin and naringenin reached equilibrium. So we set 12 hours to be the incubation time. According to the results, we could see in this table, the protein binding ratio of naringin was the highest in rat plasma, and the moderate in human plasma, then the lowest in dog plasma. And for naringenin, it is similar, and they are all over than 93%. So the differences of protein binding between naringin and naringenin indicated that the glucose side of naringin was the most important factor in the process of protein binding. So in the serum albumin, only a part of plasma protein could not reflect the whole completed plasma protein. So we found that in some previous research, the binding contrast of naringenin to human serum albumin was more than 4 times than that of naringin. So that is to say, other protein in the plasma played a very important role in the plasma protein binding of naringin and naringenin. To novel drugs, metabolites would show the metabolism profile of a certain drug, so that we could know how the drug changes in organism, where the drug goes, and whether it interacts with the organism. After oral administration of naringin to rats and dogs at dose of 42 milligram per kilogram and 12.4 milligram per kilogram, naringin metabolites were identified by the chromatographic retention time, chemical composition, and mass fragmentation behaviors. And then metabolites were further confirmed by reference standards. So the major metabolites, including naringin, total naringenin, and the 4-hydroxyphenyl propionic acid, which was called HPPA for short, in the excreta was quantified by an LC mass system. So 22 metabolites and 17 metabolites were identified in dogs and rats, respectively. Here is this is naringin, and this is the metabolites of naringin in rats plasma. So the observed routes of naringin metabolism were hydroxylation, methylation, and so on, are shown in this table. On the basis of these identified metabolites, a comprehensive metabolic pathway of naringin was proposed. About 21% of administered naringin was recovered in rat excreta in the form of naringin, total naringenin, and HPPA, and about 60% was recovered in dog excreta. The level of HPPA in excreta was higher than those of naringin and total naringenin, and the quantified metabolites were excreted more through feces rather than urine. Most of these metabolites were excreted within 36 hours post-dose. After the oral administration of naringin, the major metabolites found in blood was naringenin-glucuronic. In our study, 2 of the glucuronides, here, number 9 and number 10. 2 of them have also been detected in urine, feces, and rat bile. except for the acetylation positions of acetylate naringins due to the characteristic product ions. The conjugate positions of the metabolite were unknown by the UPLCQ-TOF method. However, because of the low levels of metabolites in the samples, it is difficult to enrich them for NMR analysis. So in this study, most of the reported phenolic acid metabolites of naringin has been found except HPPA, and it is presumed that all these phenolic acids were produced by different patterns of bond fissions on the C ring. This is the C ring. And subsequent oxidation. So, and this is the profile, the possible profile of naringin and naringenin in rats and dogs. Okay, we also studied the metabolites in rat and the human liver microsomes. So in this study, most of the reported phenolic acid may be found, except HPPA, which I just mentioned. And this is the skeleton of the flavonoid compounds. According to the relevant chromatographic peak area of metabolites in excreta, naringin-free naringenin, naringenin conjugates, and HPPA were suggested that major metabolites in vivo. So the duration of urinary, the fecal excretion of these major metabolites was studied after an oral administration of rats and dogs. So let's begin with the last part. Enzymes catalyzing the metabolism of drugs play key roles in liver, which are abundant with enzyme systems, especially cytochrome P450s. And it is a family of heme-containing enzymes involved in the biotransformation of xenobiotics as well as endogenous compounds, playing an important role in drug metabolism. In FDA guidelines, these isoforms are very important and should be and these isoforms could metabolize about 90% of the merchant drug. So we choose five of them. Actually, it's 7. We just tested the other 2 after I submitted the abstract. Okay. So citrus fruits and grapefruit juice have shown to contain coumarins, furocoumarins, flavones, flavonoids in both the free and glycosidic form, in addition to abundant of naringin, bergamotin, dihydroxybergamotin, and naringenin. So citrus fruit and the grapefruit juice have been demonstrated to cause the drug interaction, altering the pharmacokinetic parameters of various drugs, shown in this table. So bergamotin, I just mentioned, one of the main components in grapefruit juice, inhibited the drug metabolizing enzyme activities in more than half for CYP450s, 2A6, 2C9, 2C19, 2E1, and 3A4. Some are not shown in this figure. At a concentration of 10 micromole using human liver microsomes. So the interactions between naringin, naringenin, and P450s are still not clear yet. So in the present study, the inhibitory effects on major human CYPs bynaringin and naringenin are investigated using human liver microsomes. So this is the IC50s of these isoforms. So IC50s were measured respectively. These results demonstrate that naringin and naringenin display a weak inhibition for CYP2C9, about 30 and 40 for naringin and naringenin. We also investigated the induction of pregnane X receptor by naringin and naringenin. It is a member of the nuclear receptor family of ligand-activated transcription factors that includes the steroid, retinoid, and the thyroid hormone receptors, so as well as many orphan receptors. PXR regulates the gene expression for many drug metabolizing enzymes and transporters. So until now, the PXR induction effect by naringin and naringenin has not been reported according to our updated knowledge. So in our study, 10 micromole rifampin, the positive inducer, induced PXR mediated luciferase fluorescent increase of 3.3-fold with comparison of the control group. Oh, sorry. So naringin and naringenin displayed about 28.4 and 16.5% of rifampin-inducing PXR-mediated CYP3A4 transcription in HEPG2 cell lines. These results display no significant PXR induction potential for naringin and naringenin. After studying the inhibition and induction, the effect of CYPs towards naringin and naringenin were also investigated. In order to make the metabolic pathway clear, we studied all the enzyme metabolized naringin and its aglycon. 6 metabolites were chosen to be quantified. We used correlation analysis and chemical inhibitor to do the job. So this is the figure of 1A2. And for the correlation analysis, use the production of selective substance metabolites as the x-axis, and the production of naringin's metabolites as the y-axis. So if the metabolites was metabolized by only one isoform, the points would be linearly associated. So we couldn't see linear association in the 5 isoforms. And we also used chemical inhibitor approach to test this. And the metabolites was demonstrated to be metabolized by multi pathways, and the figure showed the percentage of metabolism. So finally, we could know metabolic pathways in this figure. Yeah. So enzyme of phase 1 and phase 2 distributed widely in organism. Other tissues and organs could also have a certain amount except liver. In our study, metabolites were detected in the excreta of rats in the urine. So kidney may be an important organ in the metabolic process of naringin or naringenin. We found that in rat kidney microsomes, phase 1 metabolites could rarely be found, but the conjugation of naringenin and glucuronic acid were found easily. So we used the Q-TOF to identify the metabolites, and compared with the mass spectrum of standard of conjugation. Finally, we validated the structure, and kinetic studies also reflected that this compound could be metabolized by excreta very fast and would not accumulate in the organism. So, at last, we have got the clinical trial permission of China Food and Drug Administration. So we hope we could provide a new idea of developing novel drugs. Okay, thank you all for your attention.